got a little bit ahead of myself when I, excuse me, ate an apple. I want to go back to the complement activation, okay? Complement activation right there. I don't see describe the complement activation itself right there. Yes, I do. The role of complement and describe the two pathways that activate complement itself. Mine is 22-14. I want to go back up a few minutes, a few steps right here, and talk to you about complement. The overall result of complement is to phagocytize the infected cell itself, okay? Now, wait a minute. To phagocytize the antigen or pathogen, mainly bacteria, okay? So we want to phagocytize the bacteria. We want to lyse the bacteria. We want to kill the bacteria. And a system that we have in place right here is a series called complement activation. Complement is a series of proteins. It's got a cascade effect. Now, let me explain to you a cascade effect. We've seen it before in a blood clotting scheme right there. But a cascade effect is, now my numbers may not be exactly right as far as what to look at here, but let's say that we have a series of proteins in our blood right here that are inactive. All right? And in the complement system, we call them the C proteins. Complement inactivated is converted to complement activated, okay, by something. That's something we will look at here in just a few seconds right here, okay? Now, this C1 active right here is going to convert C2 inactive to C2 active. C2 active is going to convert C3 inactive to C3 active, okay? I think you can see the schema of this right here. From C3 inactive to C3 active. All right? And on and on and on. But this thing goes down through C9. All right? So a complement, the two ways to activate complement. One is an alternate pathway, one is a classical pathway. Let's take a look at the classical pathway itself, okay? As we look at 22-14, okay? What we see right here is the first step to activate C1, to change C1 from C1 inactive to C1 active right here, is the recognition of antibodies attached to a bacteria. The recognition of antibodies attached to a bacteria. Now, we haven't talked about the structure of an antibody. We will here shortly. But let's just accept that we make antibodies against something, against antigens and pathogens right there. And these antibodies attach to something on the surface of the cell membrane. And the antibody attachment right there, the attachment of the antibodies right there, activates C1. Well, as you can take a look at right here, this is a bacterial cell wall where the antibodies are attached right there. There's an activation cascade. C1, C2, C3. Well, the bottom line is right here, there's a C3b that's going to be active. The bottom line is it's going to be a C3b that's activated. Now, these are inactive proteins in our blood. In our blood right now, you have them and I have them right now. We don't want them turned on, we don't want them activated unless there's a target there for them to fasten to. Because if the suckers are activated and there's no pathogen out there, they're going to attach to something. They really, really are. So we don't want these things activated unless there's an antigen or a pathogen present, but mainly a bacteria. Now, what happens when C3 is active? This is key. As you take a look over here, to the right-hand side right here, there's a big MAC attack. There's a membrane attack complex activated right here, C5 through 9. So C3b is the key to the alternate pathway, to the classical pathway, and also to the alternate pathway. So C3b right here is going to activate C5 through C9. It's a 5. Trust me, that's a 5. This is the membrane attack complex. This is the membrane attack complex. Membrane attack complex. This is going to put pores, create pores, holes in the surface, in the cell membrane of the bacteria right there. Water runs in, the cell swells, the bacteria cell swells, and it lyses, okay? Seems like something simple, doesn't it? Allow water to come in and kill it. This is the classical pathway. The alternate pathway right here doesn't have all these steps right here. There's a protein called perforin. wait a minute a second right here, propyridin, P-R-O-P-E-R-D-I-N, I knew it was a P. Propyridin is activated. Now this is kind of by accident right here, this alternate pathway right here. Propyridin activates, interaction activates C3b. So this is kind of an accidental pathway right here. So there's an alternate pathway that does not involve the attachment of the antibodies to the bacteria right here. An alternate pathway activates propyridin, activates C3b in the alternate pathway. C3b is going to activate the membrane attack complex and create pores in the bacteria itself. Now, probably 90% of the complement is going to be the classical pathway. Probably around 10% is going to be the alternate pathway. That's my guess. Okay. Now, with that in mind, let's turn to our focus to the last objective on the front page right here, the MHCs. I want to talk to you about MHCs, then we'll shut it down for a while. All right, MHCs. All right, let me get rid of this right here. This is a complement pathway. Now, MHCs are proteins. Genetically transcribed and translated proteins. Chromosome 6, I think, but that's not important. I'd have to go back and look it up right here. But there is an MHC, oh, what the hell? There's a MHC protein. Let me get over here. Major histocompatibility complex. Yeah, it's not something that I think of every day. Major histocompatibility complex, MHC. We're going to focus on MHC1 and MHC2, okay? Now, MHC1 is produced by all nucleated cells. All nucleated cells can activate and produce this protein called MHC1. MHC2 is produced by antigen-presenting cells. Antigen-presenting cells. Well, what kind of a cell is going to present an antigen? Well, those are our phagocytic cells, okay? APCs are going to be on monocytes and microglia and the Kupffer cells, the macrophages in certain tissues. So they will present the MHC2, all nucleated cells will present MHC1, okay? Now, let me talk to you a little bit. Let's see, we'll go back and take a look at this right here. Describe the role, turn your page, describe the role of two CDs, include cellular response. No. Let me go back. I got to talk to you about MHC1 and MHC2. Figure 22-18. Figure 22-18 has the antigens and MHC proteins right here. Let's take a look at MHC1s right here, okay? MHC1s. Now, we mentioned earlier about an antigen being presented on the surface of a cell membrane, or on the surface of an infected cell. Like the role of interferon is to interfere with viral replication in adjacent cells, all right? But here we have a protein from an infected cell that's going to be presented on its cell surface. It's going to be recognized, and this cell is saying, kill me, kill me, kill me. I'm infected right here. So here's what happens right here. We have the surface of a cell membrane, and we have a virus attached, okay? We just put a little virus, a little protein code out here. That's a virus. That's a receptor site for the virus right there. The viral protein, let me back up. The genetic material from the virus, this coronavirus, this RNA, enters into the cell. I'll make this RNA virus right here. Here's the cell. All right? Here comes the viral protein. Get me? Here comes the viral genetic material. Now once this genetic material is in there, if that dadgum thing just goes into the nucleus and is transcribed to the DNA and sits on our DNA, it's going to be hidden. But this genetic material right here may be processed, may be fastened to a lysosome. And that's what we see right here. We have a plasma membrane, viral or bacterial pathogen right here. This is a virus right here. If it's all nucleated cells right there that have MHC1, maybe it's a bacteria that entered in it. But irregardless, we're going to process this abnormal protein, abnormal genetic material in our cells right here. So this is a viral RNA for the coronavirus right here. More than likely, it hasn't been activated yet. Okay, so here we have this thing forming up, marrying up, attaching to a lysosome right here. And here we're going to process this right here. May not be the best story for the coronavirus, okay? So probably a better story for a bacteria that has infected a cell instead of a virus, especially the coronavirus. Don't know that much about the coronavirus, not yet. Okay, so here we have some ingested material, foreign material right here. It's going to be processed with a lysosome right here. These fragments that are going to be processed, digested right here, are attached to an MHC. Processed, MHC, processed. No, I'll put a little fragment up here, okay? I'll do that. I'm going to put a little fragment here. Here's a little fragment right here. Here's the fragment presented over here to the cell surface right here. Again, it's a better story with the bacteria that's engulfed, that's been engulfed, that's entered into one of our host cells right there. Been processed with a lysosome. Fragments of the processed bacteria are presented on the cell surface by an MHC1. Now, this is abnormal. It's abnormal to have an MHC1 on the cell surface with anything attached to it. Okay? Now, an MHC2 is going to be on all antigen presenting cells, the monocytes, those fixed macrophages that we talked about right there. And here we have again, figure 22-18 right here. Here we have an engulfed bacteria. Engulfed bacteria married up with a lysosome right there. Oh, well, often we do have lysosome action, produces antigen fragments right there. The fragments are presented by